For most of human history, biological manufacturing was a process of observation, finding what nature had already built and learning to harvest it. Today, we have moved from harvesting biology to authoring it. This began with biomanufacturing 1.0, characterized by the refinement of fermentation. When Chaim Weizmann industrialized the ABE process in the 1910s, he wasn't inventing a new function. He was using the natural metabolites of cells to produce acetone and ethanol at a commercial scale. As the field progressed into version 2.0, researchers began capturing secondary processes, the complex chemicals cells create for defense. While these defense mechanisms were ideal for antibiotics, the industry remained restricted to the specific molecules nature chose to produce. Alexander Fleming's 1928 discovery of penicillin is the landmark of this extraction-based phase. He identified a naturally occurring mold that could destroy bacteria, using technology to unlock a potential that already existed in the natural world. This classical approach revolutionized medicine, but it carried a biological ceiling. Scientists could only discover and scale existing functions. They could not yet design them. We are now entering biomanufacturing 4.0, a shift into what is often called biotech 2.0. This represents a transition from discovering natural molecules to the de novo design of entirely new ones. Researchers are no longer searching the natural world for biological answers. They are actively writing new biological blueprints from scratch to perform specific, engineered tasks. In this paradigm, biology functions as a rigorous engineering discipline. Design-build-test loops have replaced manual screening, requiring a complete overhaul of how we handle biological information. This new architecture is defined by a single continuous pipeline, the digital-to-biological loop. To understand how the modern bioeconomy functions, we have to look at how this loop translates digital code into synthetic living systems. The catalyst for synthetic biomanufacturing was the rapid digitization of the genome. Human genome sequencing costs have plummeted. In 2001, it cost over $100 million. By 2026, it will cost roughly $100. Compared to Moore's law for computing, our ability to read DNA has vastly outpaced processing power. Cheap sequencing allowed us to convert physical DNA into a digital language. Once genetic code became data, it could be stored, shared, and simulated in cloud environments. By making the reading of DNA essentially free, we unlocked the ability to begin writing it. In the design phase of the loop, genetic networks are engineered entirely within computer-aided design software. These digital simulations allow scientists to predict protein folding and optimize genetic vectors. Once validated, the digital sequences are sent to physical machines for the print phase. Older chemical methods are being replaced by enzymatic printing. This uses TdT enzymes to assemble DNA base pairs one by one. By removing the constraints of traditional organic chemistry, enzymatic printing significantly increases the speed and accuracy of DNA writing. We have successfully decoupled biological design from physical labor. DNA can now be printed with the same precision we expect from digital manufacturing. In the biomanufacturing 3.0 phase of the 1970s, researchers learned to insert single genes into E. coli to mass-produce insulin. This proved that we could trick bacteria into making human proteins. However, modern synthetic biology requires building much larger molecular assemblies, which means multiple DNA fragments must be linked together without errors. Gibson assembly solved this by using an isothermal process to link overlapping DNA fragments into a single continuous loop. This control over DNA assembly means we are no longer restricted to the standard A, T, C, and G nucleotides found in nature. Xenobiology researchers are now integrating artificial nucleobases like D56 and DNAM to expand the data storage capacity of the genetic code. In transcriptomics, we are using chemical alterations like pseudouridine to modify messenger RNA, allowing synthetic sequences to bypass a cell's internal immune defenses. By reprogramming the machinery of the cell, we are creating biomaterials and proteins that the natural world never produced. While CRISPR was a breakthrough, the Cas9 nucleus acts by creating double-stranded breaks in the DNA, which can be a blunt and unpredictable way to edit a genome. Base editing is more precise. It chemically converts one DNA base directly into another without breaking the overarching strands. Prime editing acts as a genomic search and replace, finding a specific sequence and accurately swapping it for precise insertions. These tools allow researchers to scale their work, moving from editing single genes to constructing entirely synthetic chromosomes for complex organisms like yeast. Genetic modification is becoming a high-throughput process of algorithmic code execution. To apply this loop at a global scale, the industry has moved into the biofoundry. These facilities use lab-on-a-chip arrays to automate the testing of millions of synthetic designs simultaneously, using microfluidics to move material at high speeds. Machine learning acts as the logic engine for these foundries. Models like AlphaFold predict protein structures in seconds, feeding that data back into the CAD software to refine the next generation of designs. We can see the results of this loop in cellular agriculture, where companies are now growing meat products from animal cells in a lab, bypassing the need for livestock entirely. In agriculture, this loop is producing crops with targeted resilience, such as these plants engineered specifically to thrive in high salinity soil. The biofoundry represents a self-improving system where AI design and automated physical testing constantly accelerate each other. As biology becomes an information science, it inherits the vulnerabilities of the digital world. The cloud integration and massive computing power required for the digital-to-biological loop make biofoundries a prime target for cyberattacks. While the theft of intellectual property is a constant risk, the vulnerabilities in synthetic biology extend to physical infrastructure. These are cyber-physical threats, where a digital breach is used to physically interrupt an assembly line or corrupt a fermentation process. A malicious actor could subtly alter a single line of digital code in the CAD phase, corrupting a synthetic organism before it is ever printed. We are writing the future of our food, medicine, and environment in digital code. Engineering robust cyberbiosecurity is no longer a secondary concern. It is a fundamental requirement for the synthetic loop to function safely.